Okay, we're in Zoom now. Muted.
And so I'm trying to let everyone know their numbers in the next payment that they owe because the next payment's due October 22nd. So if you know short term,
Nobody's answering here, so
but like I can't just kind of wait and wait because then what
why is
happens
this
is
not
they're
going
having to
away?
send money to me. I would have to like work on
did.
contacting people. So the sooner
Now
you
it's
know,
gone.
the better.
Maybe I just
So I think we have space and I'm
right way.
gonna
Okay, so good to go. It's
Hey!
unmuted. It's recording.
Cool.
Lots of blame. Oh, great. That'd be perfect.
Stuff.
Yep.
Otherwise,
It rained.
very
That's
good.
okay. Yeah, umbrella photos. It is what I don't think
This is ours, probably going to introduce you.
If he wants.
Yeah, so it's yeah, it's a whole house because there's like 11 or 12 of us.
What time do you have it there?
10:56.
Yeah, it's nice. And it's by the lake. I know, me too. I'm really excited about this. I think it'll be fun.
I'm daring to see your notes. Do you care? I think I have any notes, so you don't mind.
No, yes, there's only 3 participants right now.
Because if you swap, then this is going to be like that, and then you have to write. But I don't know if Chloe's coming. I can play with it, but I don't know. Do you remember, like, I know Chloe was doing it, but there is something I know how to do. Do you care if you have presenter mode at home? So do you need it?
No.
In which case, you could just swap them.
So I just swapped them.
steady it.
Well, not about swapping, but like you could just cancel presenter mode in general.
Yeah.
But like, there's a lot of crossover where I'm not too worried.
It's kind of helping, but also great. I thought we were gonna have to like break it down like that. But he's like more relaxed because he's like, you do combination like
But it didn't swap them, you just cancel
That's gonna be a lifesaver.
Okay, it's just showing this. Okay, and I think so.
print out all of the keys.
Yeah, that's what I
Is
think.
that the one of the keys for chronic? But to lay them out.
Just ask them if they can see the full screen.
Yeah.
Are y'all
Or you can ask them.
in hearing us? Hi everyone.
Yesterday I was looking at one. And I was like, oh, I guess it doesn't have antennas.
Cool. Alright, it sounds like they can hear us
I
and
know,
see us.
the antennas just fell off.
They can see and hear.
Yep.
Also,
They can at least see the screen. I don't know if they can see me or not, but
Yeah, let me, whenever the seminar is done, I will get on my laptop and I'll send you the, like, ballpark range numbers. But yeah, it's pretty cheap, so
I'm gonna do tonight's trip, which I'm excited for.
I'll do a mod. I have some labs, but it makes it sick. Big ones are variety.
So, please take your seats. We're going to get started. We're going to get started. Please take your seats. So, it's my pleasure to introduce today's seminar speaker. Is Chase Stratton. He's coming from Delaware. He's right now an assistant professor in the Department of Biological Sciences at the Delaware State University. So I know Chase for quite some time. Like, I think we met 10 years ago when he was doing his PhD in at the University of Vermont. He was working with Yolanda Cheng. So I went there. He was working with Sweet Mitch at the time, looking at essential oils and repellents for the Sweet Mitch. And we have this interest on repellents, attractants, so he was looking at push-pull systems. So after finishing his PhD in Vermont, he went on to do a postdoc at the Land Institute, and then after that, he got his position in Delaware. So I think he's going to be talking mainly about the work that he did at the Land Institute with essential oils and thrips. So welcome, Chase. Thank you for coming.
thank you for having me. Hi everyone. How we doing? Semester going well? Anyone got a bunch of exams this week? Yeah, we're kind of in crunch time, huh? Research going well though? 'Cause that's what I really want to talk to you all about, whatever you have going on with insects. So for this talk, I'll be sharing some of the work I completed as a postdoctoral researcher at the Land Institute. So for those of you who haven't heard about the Land Institute yet, they are basically trying to breed perenniality into food crops or other we're trying to get a more permanent agriculture. So at the end of harvest, you don't rip out. The roots keep growing, the soil stays intact around the plant, and then the next year it grows again and you harvest again. So that's that work, or at least what brought me there. I was doing sustainable pest management research, and I'll get into that trajectory. But so this is my current lab logo, if you will. I kind of just took Delaware State's, but I focus on plants, insects, microbes, and these days it's more data science and how to manage and use data for interpretations and asking questions. That did not progress. Okay, so just to give you a general framing of how my entry into agricultural research got started, we use a lot of inputs to do things that plants do on their own. And we have to feed a lot of people, we have to produce a lot of food, and basically we're putting these non-natural alternative molecules in the systems to try to make sure we're getting food and defend against pests. But there are aspects of plant ecology that we should reincorporate and can reincorporate to help plants defend against pests or other pressures on their own. And specifically what I'm going to be talking about today are plant essential oils. And so every single plant has its own unique chemical profile, if you will. And there are certainly compounds in general that they have in common across different species, but the whole profile of plant chemistries for each one is unique. You could think of it as a fingerprint. And plant essential oils, has everyone heard plant essential oils of these days? Yeah. So they're extracted by various methods. The typical way to get these low weight molecules is called steam distillation. For anyone who's wondering, you just take the plant tissue and cleanly break it down with steam and basically condense the chemicals back into a mineral oil that can be applied. There are tons of claims of human activities. They're really good at killing insects, these plant defensive compounds. Many of them do have potent biological activities in humans, but a lot of it is exaggerated these days, that potency. And we won't get too far into that. But this is a good group to discuss the initial thinking that went behind some of our predictive space in testing essential oils. And so on the right, you see data that were generated at y'all's ag station, I guess around 9 years ago, on one of y'all's GCMSs. Actually, no, this was GCFID. So gas chromatography, flame ionization detection. You don't get the chemical name per se. It doesn't have the mass charge that you need to get that structure, but you do see the different masses that come out. So there's a long coil. It heats up whatever chemicals you're testing and separates them by mass. That's the gas chromatography technology. And so by putting it on this long length of coiled conductive metal, you can manage the temperature precisely, and basically you know masses of this range are hitting a detector at this time. And with the retention time and those masses, you get these areas that, these are all individual chemicals. So in this case, it's peppermint, marjoram, wormwood, wintergreen, thyme. We tested, I'll get into what we tested. We tested more like 18 in this case, but you can see, even though these are in the same plant family, their chemical profile is totally different. And this is actually old these days. I'm sure this group has updated their phylogeny, but this is a great resource. So if anyone's wondering about phylogenies, every single phylogeny we have right now is a hypothesis. It's hypothesized, right? We do not have the capacity yet to do whole genome phylogenies, to compare the whole organism as a genome in this data space, but this group is probably the best I've seen at having an accurate phylogeny that takes into account not just the nucleotide sequencing of the species, but also their functional attributes and trying to categorize them. So for this study, we use that to basically, what I'll get into with Sweet Mitch, basically select a diverse set of plant families. And then we look at the chemistry within it and look at the variation of chemical space across species space, okay? And we'll get farther into it soon. The key here is just showing the variation that exists in chemical profiles across plant species. And those on the right were literally taken from essential oils that we use. And yeah, so specific structures that these peaks are representative of, and you can look at them. Some of these will have shared structures, but it's important to note, like these subtle variations in chemistry, a double bond shifting over here, can have huge difference in the way it's interacting or influencing an insect. And so, marjoram, wormwood, wintergreen, thyme, lots of variation, lots of different chemicals. And these are subsets. In my own work, I've seen published research that has anywhere from 5 to over 100 chemicals in one plant. And don't even get me started on how many plants we have no phytochemical information on. None. Like, collected nor anything. So why is this important for insects? Anyone want to talk about why plant chemistry is important for insects in this group? So you would know specialist insect versus generalist? So the specialist keys in, the literature talks about keying in on a plant or a group of plants that are the same species. I would argue specialists are even more only able to consume a chemical component or metabolize a chemistry of those plants. So they're specialized on the phytochemical because they're microbes or proteins that can convert this plant compound into nutrition for them as an organism are highly specific. And the way it starts for a specialist and generalist, so antennae are noses. They're the insect noses, basically. And so in that, this is the antenna. Sorry, I should be more descriptive. And these are basically pores. And so odors come in, and then there are proteins that are loose in that space that will go grab a plant odor and take it to a receptor. And if that receptor, that protein that grabbed that odor, is the right conformation at that time, it'll fire off an action potential. This is the brain. It gets processed and tells the insect, go to that thing. Right? And for a specialist, without that information, not going to that thing, right? In pest space, there could be a lot of utility there, I think. I mean, repellents, we use it for mosquitoes, DEET. We found that compound, and now it's like, use DEET, right? So the insect that we were working on at the time is this highly specialized fly, tiny fly, that invaded from, originally it was Eurasian origin, and then invaded Canada, and then through, I think it was Niagara area, came to the Northeast. It started in New York, and it has gradually spread. It was in Vermont while I was there. We published in 2018. I guess like 2014, it had started really establishing, and it's a specialist of brassicas, which are like the best vegetable. Anyone want to challenge me on that? So broccoli, cauliflower, kale, Brussels sprouts are all the same species, all Brassica oleracea, and this one's specialized on them. And so in our early work, we figured out a way, we developed a method to basically apply known quantities of their larvae, their offspring, to growing points. So their biology, they basically, female puts, well, after mating, puts an egg, clusters of eggs, on the meristem, the growing point of, say, broccoli. Broccoli is a heading brassica, so that growing point is the marketable portion of that crop. You need a big, lovely broccoli plant, or no one's going to buy it. No one's going to buy this, right? And so the eggs are laid on that growing point, the larvae hatch, and basically throw up on it. They're throwing up enzymes that tweak the, well, I can't go too far into the physiological disruptions that they do. It's still in exploration, but they're proteins that are messing with the growth programming of that meristem in various ways. Hessian fly is kind of the species that we were tapping into to think about what could be possible here. And it's wild when you get into salivary proteins and the chemical structures that, with Hessian fly, I maybe shouldn't share this, this might be proprietary. They have this protein that is coiled, and it basically, it's like a spring, and it pinches the cell walls. So then cells next to each other spill into each other. And that's no longer able to defend itself as a unit. It's a big pool of resources for them to slurp up, basically. And that's how that protein challenge works. But I digress. We found one larva with this method, half the time was rendering produce unmarketable. Half the time it would lead to just no broccoli, or something like this. from that tweaking the programming of how it grows. And so repellency seemed like a great direction forward for this crop. Break all contact between that female, mated female, and whatever you're trying to protect. And so the methods in this for sweetenage work started pretty straightforward. We caged plants, put basically one of 18, no, I think it was more like 15 essential oils on the plant, put females in there, let them, well, we put females and males, let them mate, let them oviposit, and then count the number of larvae that they're putting on each plant. And I brought up the phylogenetic and the chemical components, and what I want to tap on here were some of the methods for the phylogenetic, which these are all established. There are bioinformatic pipelines to do phylogenetic analysis these days, and if anyone wants to really talk to me about what that looks like and how to do it, I'd be happy to. But just briefly, we're using conserved genes. So in this case, chloroplast genes. You want to use, when you're studying evolution of species, or when they're popping out in evolutionary time, you want genes that are stable, that aren't changing quickly. So something like an odorant binding protein that's dynamically responding to chemistries as they're changing, you wouldn't want for phylogeny. Chloroplast genomes, maturase K, and rubisco large, the large subunit of rubisco, so they're involved in photosynthesis. And so it's this shared trait that is takes a long time to change. And that's how you get this basically phylogenetic distance. You can quantify when things started diverging, right? And at this point in our thinking, and at this time in the literature, the chemical space on that was still being explored. Like, how do chemistries, plant chemistries, vary and diverge along that evolutionary window? If you think about it, these genes, you know, they're involved in the energy transfer. But those dynamic chemical spaces are a series of enzymes, chained enzymes that break components apart and put them back together. They're very dynamic. So you can have totally different chemicals, but very similar phylogeny histories. You can have very similar chemicals and different phylogeny. There's a difference between the chemical variation and the phylogenetic variation, and it hasn't really been defined yet, is what I'm getting at. So in this study, we found that both of those predictive variables, phylogeny and physicochemical similarity, so the structure of the chemicals compared to each other, that's something I won't tap too far in how we're doing those comparisons, but we will talk more about chemistry and how to use it for these sorts of predictions of repellency. But we used it here and we found they were both relevant, but we eventually, I moved on to a postdoc at the Land Institute, and I continued that chemical component because the chemical component seemed very important. So basically here, we're furthering this predictive framework, but more so focusing on shared chemical constituents of plant essential oils and assigning mortality effects to those using principal components analysis. And to start, we have a newer method of high throughput screening with just plant leaves. With Thrips, it made it a lot easier. You can put them on the plant, and they're not really going too far away from this. But we have a team that does basically the screening and then mortality tests, team that does the mass spectrometry, chemical stuff. They're on Shimadzu with different technique than I'm used to, but and then we have the statistical framing to do the principal component analysis and linear regressions. And so just to run you through the methodology here, we pick the candidate essential oils, and then you just run through a series of applications where our species here was Thrips, and they were on alfalfa leaves. And then you apply the dilute essential oils. These are purchased from doTERRA, for anyone wondering. And then you give them some time and record how many of say we applied 10 thrips. How many are alive after 24 hours? How many are alive after 48 hours? And in some cases, we did find, which is a different direction that I'm still thinking about. Some essential oils are phytotoxic to some plant species, but not all. So think about that, this chemistry component. And herbicides, right? Natural herbicides. So in this, basically we put it back through the statistical test to come up with top candidates, and then to test them further, this is just a single leaf assay on it wasn't a petri dish, just a small container. And then these we went with a whole plant and test the top essential oils along with well-established flunicamide pesticide for thrips, just to show which does better or which does worse, or have a comparison. And then of course we have a water control. And so basically, essential oils performed really well. This is in the single leaf assay, and you can see, with the exception of like eucalyptus, which had similar strange effects in sweet mitch. Not the same as the other essential oils. It didn't seem to repel sweet mitch, which I'm just remembering right now actually. Ginger did okay, but you can see oregano, cassia, thyme. Oregano and thyme are usually up there for essential oil efficacy. So basically, we take the top candidates from this, especially the ones that weren't phytotoxic, and we test them in the whole plant assay. And you can see every single essential oil performed as well or better than the pesticide, and that's flunicamide. And yeah, interestingly, they're all doing just as well. But the interesting part about this, in my mind, is developing it further along these predictive framings. And so with principal component analysis, you can start to dissect the individual components of the essential oils and those chemicals that are drawing certain treatments toward mortality and certain ones away from it. And basically, if you look at the paper online, this is some of the information you'll see published on it. And we found, I think, 14 individual compounds were significantly associated with mortality effects. And with these linear models, linear regressions, you're seeing, and this is on the PCAs, the PCs, you're seeing alpha terpenine, beta caryophyllene, terpenin 4-ol have positive associations with mortality, meaning essential oils that have higher concentrations of those are killing more of the thrips more consistently. And while 1,8-cineol was the opposite. Any questions? Yeah.
Of the oils you tested, which ones were phytotoxic?
Yeah, the ones that we had that asterisk.
Okay.
Yeah. And oregano, actually oregano was not phytotoxic with broccoli. Cinnamon was, and cassia was. Yeah. And that's something that I'm totally interested in, applying these as herbicides. No one's doing it yet. And even like this same framework we're doing for insects could be applied in that context too. Just trial them against different plants, right? And I want to point out, flunicamide, Thrips is resistant to already. They're resistant to like everything. Western flower thrips. Has anyone looked them up? Pesticide resistance? It's crazy. They just keep throwing it up. So, I digress again. Any questions about this methodology? Because I'll put you in touch with our stats guru, who Ebony is a whiz. But I'd be happy to talk through this level of thinking here. Yeah.
No, I'm not a big stats person, but the 1,8-cineol, the x-axis is really going from 0 to 75, and there's not much in between there. Can you explain that?
Give me your email and I'll put you in touch with Ebony. But I'll bring it up. Yeah, I'll ask her. That one did throw the results off, and that could be fine. Yeah. Could just be a factor of it's not present in many. It's in eucalyptus. It's interesting. Thank you for bringing that up. Any other questions? Please do. Yeah.
You mentioned that cinnamon also works against the thrips or the flies.
Cinnamon burns everything that I have seen. It melted broccoli plants.
So that's why it's not on the potatoes or anything. Or
Well, so for this research it was. But anytime I applied it to broccoli, it just melted. Like I put acid on it. Like literally, they turned brown. The leaves are just gone. I don't remember if we tried it with alfalfa, but it does share some chemicals with oregano, I think. And that's kind of the space we're in. So these are focusing on, the shared chemicals, right? So it's analyzed across. You have to make sure that you're able to do this. You're able to take a step into that stat space, but then you could do these regressions across individual chemicals. And we could look at these trends in those other test cases as well. So
I think you're right, Chase. I think the signal might be present in one species or one of the oils that you looked at.
You're totally right.
And it caused a different effect.
Because we're trialing that one, and this one's definitely proprietary, so I can't share that much. But in a different solution space, and it's having different results.
So the terpene, the caryophyllene, terpineol, those are repellents. Like that trend shows repellency, whereas cineol shows attraction.
Okay.
I guess.
Yeah. Sounds good. Any other student questions? Yeah.
Oh, not student, but still a question.
Right. Cool.
Your population of thrips that y'all were testing, you said there's a lot of resistance to insecticides. Did you have a known susceptible population that y'all were testing?
These are just from the greenhouse.
Okay.
Yeah, we kind of had to do with what we had.
Oh, yeah.
And I would love to think more about the history of their treatment. They're doing a great job at TLI's, the Land Institute. They've been using essential oils for management for years. So they kind of had to stop managing to get the population back up so we could run these tests. But yeah, they weren't usually applying, the flunicamide was something we identified from research, but I'm pretty sure they're resistant to it, just in general. But yeah, you need to look at that population. Yeah. All right, any more questions? Because I'm about to switch it up so we can talk future, yeah.
You said that they had to stop the essential oils so that the population could grow. Was there any that they had to just take off the list entirely because it was the thrips were already resistant?
All of them were somewhat effective, at least compared. Well, eucalyptus, we didn't include eucalyptus and ginger in the next study in the whole plant because they didn't show the same results. Yeah, and we also excluded because if it's doing more damage than the pest, you're not going to use it for management. Yeah.
I think it's like 100% essential oil extract.
Good question. I should have put it up front. No. 0.08.
just to kind of understand that dilution and what it looks like compared to a single plant or something like that, or a row of plant. What kind of potency is that comparable to an actual just plant?
Yeah.
Because there's that whole trend of everybody's oh, I'm gonna put this lavender plant here and it's gonna protect me from all the mosquitoes, and it's well, first you gotta toss it up a little bit for it to start smelling. But I mean, how much do you need of how many rows of lavender is that 0.08 dilution that you're using?
Yeah, that is a crazy cool calculation that could be done. You can look at the volatile rates, but these aren't just volatiles that we're extracting in essential oils. So plants in odor space are releasing low weight molecules all the time. all the time. Some of them are signaling to, like a natural enemy, come here and help me. Some of them aren't. Well, none of those volatiles that I know of, I mean, I'm sure there's some, aren't like acutely toxic, aren't making contact to melt unless it's directly applied on them. So even if the dilution is the same, it's making contact, right? For repellency, it's that volatile hitting the antenna. But mortality, in this case, for a greenhouse pest, you're spraying it on them, right?
Is there some sort of like an oil that attaches to the volatiles so they stick there, or?
Well, so in this case.
Oh, soap. There it is. Sorry.
Yeah. In this case, you just put a little drop of soap for this, and yeah, they're in mineral oil, so they don't mix with water unless you have that soap, and that soap also helps it adhere to plants. Yes.
So for all of those essential oils, you're using a 0.08% dilution. How did you come up with that specific calculation, and why isn't there like variations in like dilution for like the different?
There should be.
Okay.
Great question. This is, well, we settled on a number that was representative of the company's recommendation of dilutions, basically. But yeah, you should do a serial dilution and find. I'm in the realm these days of, okay, let's pick out all of the ones that were positively associated with mortality, extract those individual chemicals, and put it into a blend, and apply that. Repeat your question, actually.
No, I was asking why there isn't like why there aren't like different levels of like dilution for the essential oils based off of like what's most effective for each.
Right, and I think that's future work. Really important future work. Totally. Yeah.
For the pesticide you used, is there a specific reason why you chose that one?
It's just commonly used.
Okay.
Yeah. Yeah. And I should have looked up, I mean, we could right now look up the active ingredient and look at the structure compared to other stuff. I don't know what it targets. I won't look it up right now. But we could talk further about that, because pesticides, their active ingredients are targeting specific things in many cases, or like the general layer, bilayer, or a protein that's embedded in it, or a neuron, or a family of neurons. So.
I think there's a question on the chat.
Oh, great. Oh. Yes, I will do so. My apologies. Yeah, so many good questions. Is any of it recorded over there? Like, or is it just lost to? Are y'all just aren't? Okay. I'll do a better job repeating questions, because I want to take this into the next space. Well, let's finish talking about this. So, in chemicals, structure defines function, right? It's not going to interact with the thing. It's not going to cause that function. And there are ways to compare structures, chemical structures. So basically, we need to do this in more cases. We need to study more insects. We need to apply it in different contexts to see what's going on where. And these are published methods that are accessible for anyone. And I just, the variation that exists in plant chemicals and what they could or couldn't do to insects of different species fascinates me. And so a project that we're going at this point is in artificial intelligence. Who's privy to artificial intelligence? Is everyone, when y'all hear it, what do you think? ChatGPT? Yeah, so this is newly NSF funded work. We're planning basically to use machine learning to train a neural network. And that neural network will be based on, in my mind right now, the neurophysiology of fruit fly brains. It was recently published. Look up fruit fly brain architecture, neuron to neuron connectivity. And basically, so the idea is to set up these recordings of insects responding to, I say recordings, but it needs to be converting what's going on in the container. So you put, say, 1,000 thrips. Well, for us to start, we're doing Drosophila melanogaster, fruit flies, because of all the tools available in genomic and neural space. Apply these odors, one of, well, however many, and see what happens. And I say see what happens, but really it's the machine seeing what happens, right? And so you're applying an odor, 1,000 fruit flies are in there. What happens? Until, in my mind, until the end, where everyone that's in that container has gone through every series of steps of what they're going to do in that context, right? And so with those information, we can start programming these behavioral variables that the machine will start to be able to learn and interpret. And so in my thinking, since we're using it for pest management to start with, the behaviors I had been thinking was time of first twitch. Like in my pest management research, I've seen specifically with like cinnamon in an olfactometer, the insect is stressed at first, and there's finite behaviors associated with that stress that in this case are important, right? So once we've trained that neural network, once it has learned every finite detail of, you know, this level of insect behavior, this is not yet generalizable, right? That's what the goal is. You want a neural network or a brain, a digital brain that can observe behavior in the field, observe an insect doing something in the field, and apply a molecule to change its behavior, right? And so having eyes on every finite detail, every interaction in that container is now something that the digital brain can reason with. And, you know, Drosophila melanogaster is the start because we have genetic tools, we can alter a protein. So, as an example, I briefly touched on the agricultural impacts, potentially, of having so it's A-insect is what it's called, is the pronunciation, A-insect. Not just being able to apply the right plant chemical at the right moment to get a pest away from a crop, but in medical, as a medical tool, knowing the genome, knowing the neural connectivities, we could develop basically a model digital brain for testing next generation medicines, basically. So let's use CRISPR to put a protein or a gene that produces proteins that cause Alzheimer's and Drosophila. And we already have it trained on healthy, normal behavior. And this neural network has access to that published single neuron connectivity. So aberrations in behavior can be assigned to neurons in that space. And so it allows you to test next generation medicines that get the behavior back on, right? And that's AI space. That's tapping toward it, right? But, so you gotta give it a starting point that it can go off of. Like, you have to verify to the machine that that time that you were thinking the first insect was stressed out is actually the time it was happening. You have to confirm that visually. So I'm thinking about these behavioral variables that set off an easy way to confirm that that's what's happening there, that's what's happening there. And I want to reach out to you all. Another group that I talked to brought up other test spaces. So I'm still in the simple Drosophila melanogaster and all the tools we have available about it, responding to chemicals. Well, plant chemicals. Another group, and you know, it could be Hessian fly, it could be Swede midge in here. The more insects we study, the more generalizable it becomes on behavior across species, right? And one group even brought up, how about predator-prey interactions, eventually, right? Branching out into being able to interpret like ecological interactions and using what we know about these insects and building the rationale behind what's going on, right? So, anyone want to talk about variables that we should include, baseline? Like, it could be as finite as, you know, hair on tarsi moves.
Well, what about, like, grooming of themselves? Because if an insect is stressed out, it would probably be more likely to groom itself, trying to get whatever volatile off of itself.
Yeah, yeah, see it all the time. Cleaning antennae. Yeah, yeah, totally. It's in the old brain box. Yep, yeah.
Could be that insects also emit different odors, so maybe there is something that they would emit from smelling something else.
I love that. So, this is an airbag, an SPME, solid phase microextraction. The idea is absolutely collect everything that's coming out and take regular samples. So I have a software in R, well, it's a package in R, patent pending, I think I have to say that, that basically one of the functions it can do is if you know the chemicals that are being in, which we will, we'll know exactly the chemistry, it taps into PubChem and pulls published information about those molecules as it's, I mean, like, on the fly. So categorizing the things that come out is something we will have in the moment access to. And that, totally. Knowing the environment and every detail of it as those behaviors are happening is what we're controlling for here. Yeah. Yeah.
What about like distance traveled of individuals or mating of individuals, if that's affecting their mating behavior?
Distance travel is definitely something we need to. They in some cases are like trying to run away from a thing that they can't until they're exhausted. Yeah. Yeah. And it, you know, we have the. I'm not gonna call it optics because it's not technically. I mean, you know, we will be able to track individuals.
Can you like do it with like EcoVision and put it in the AI
Sure.
systems and like kind of, I don't know much about that, but
Sure.
technology really read each other.
Sure. Yeah, that would definitely work. There are a number of open source stuff, but I do the hard code. I go in, get the raw data, and build the algorithm out of it. Yeah, but totally. Those aspects are very important, and you know, eventually, the AI will be coming up with its own symbolic representation of this here moves, and then it ran for 3 seconds, and then it stopped across the whole population, and that will mean a specific thing that is well beyond my comprehension, right? But getting it to the direction that it can do that, sure. Yeah.
How about the concentration of the older components in, like, let's say, per air
I need to be repeating questions. The question is about concentration.
Yeah, what is like the concentration of the older, let's say, molecule in each airbag and can trigger their specific behavior, let's say, like,
Yeah. Yeah, so we're talking about how much of each chemical component is getting in at any particular moment. And that is stuff that we'll be tracking because we'll be putting in a dilute amount. All of the essential oils that we get are run through, it's on a Shimadzu with electron ionization. So the quantities and qualities are totally known for us. And we will be manipulating that. So once this is set up, and we're in early design stages where we're making the container, but once it's set up, the plan is to have multiple groups with a cart going and researchers running species through whatever tests. But every single concentration and variation, all of that's important. All of that's useful. But we do need to know exactly what it is. Totally correct. Yeah. Yeah.
Maybe other looking at the life stage of the insect and seeing how that affects its behavior.
The question's about the life stage of the insect, and totally. That is a space that we know so little about. We have some work going with like learning, a simple form of learning, habituation, just getting used to a thing. Did anyone see recently jellyfish apparently learn? Amoeba do habituate to chemicals. Amoebas have no neurons, nothing. And it's just getting used to a thing. So we're testing it with insects and pests to get used to a repellent, right? That was the original idea. Is we're applying this repellent, it's a false ecological signal. The ability to, logic that out, that, oh, that's a fake repellent, my host is still there, can happen. So we wanted to test, like, what's the likelihood or what's the possibility. So that's active research going on now too, but cool. I just think that in the development of that neural space, right, the plasticity involved, and exposomics is a term you should look up. And it's about every single interaction an organism has in its lifetime, right? And for plants, every single interaction. These are not just us talking, but the air on you right now. And being around other people and who they are. Like, exposomics is every single one of those details. With insects, in this context, it's every single molecule in that is influencing not only its behavior right then, but the adaptation of its neurons for future experiences, right? So that is something that AI could help figure out, especially with the tools we have for Drosophila, change and neuroplasticity and learning and adaptivity. Yeah, excellent. Keep them coming, please. This is awesome. Yeah.
It seems to me that the most challenging thing of this is measuring all these. And you're just looking at behavioral responses, right? You're not looking at physiological stuff, what's happening in the gut. You're looking at what the Drosophila is doing in response to a chemical.
Totally.
How are you going to measure a seed moving or something like that? Like, that's what I'm trying to visualize.
Yeah, the machine will. So.
You got cameras.
Yeah, the original thought was high-speed cameras, but I've been made aware of other technologies that have that for a Drosophila. I would be able to track all that. Yeah, and we're also moving toward being able to even like see the chemicals, see them in digital space. Like as they're moving in motion. So that's stuff that in raw data space with the right amount of processing power, you can bring it all together. Does that help? We getting there? Sort of. Yeah, this is an emerging space. There's a lot of unknowns, but there are so many tools available. So machine learning, right? Every finite detail of that video, the recorded video, is going to be dissected. And every single possible reason for something happening is going to be tried and thrown out or given support. And if it gets enough support, it goes into a dictionary for this is a behavioral variable that we should look for in the future.
So are you going to go through the videos as you're training and say, yes, this is
That's
this is that?
That is how you have to start. You have to verify. You have to validate. And that's why I was thinking these behaviors, and now I have a bunch more. Things that, you know, it's easy to say, they stopped moving. Yeah. And that's stuff that I'll pay students for. Yeah.
So you said that you put like 1,000 individuals into one of these containers. How are you tracking one of that 1,000? Like to see what that one behavior is. Like, so you said that like in one of those boxes, right? For one of the treatments, you have like a ton of insects inside of it.
Yeah.
So how are you tracking like?
Well, I haven't yet.
Oh, okay.
This
It
is
just
just
seems like a lot of
awarded.
chaos, a lot of insects to like be looking at the same time. Why not make it into like smaller groupings or one individual at a time?
I'm not the one looking at it.
We got people for that.
Well, people will, you know, you can have a video recording that you just circle one individual and it'll follow it for you, and
Yeah.
then that's what people are confirming.
Yeah.
The machine will have labeled every single, it'll give them all their own name if I want it to.
Okay.
It'll name all of them a new name and track them and all of those finite details because of the sensors that we have developed. And so I do want to point out, eventually, this is on a high-performance computing cluster where these trainings are occurring. It's not on a laptop. You need a lot of processing power because of there's so much information and data. And this is video, right? Well, video. So just think about those finite details we've been talking about that, I mean, I see them in like a matrix space of time where it's like this variation of these individuals are here in a three-dimensional grid over time. But looking at those patterns and trends at that scale, I'm going to tell the computer how to do it and let the computer do it. Yeah, and then we confirm by looking at one individual. And there could be other ways to test the assumptions with smaller groups. And if we find 1,000 is just too much, then we'll cut it back. This is all in like, I think a machine could do this. Prove me wrong. Yeah. Yeah.
How long do you think it would take to train an AI to like?
Depends on the machine.
To learn it?
That's a great question. The short answer is, which cluster am I on? Because, well, actually, no, with this, it's gonna, the actual training can take some time. It's more about collecting the data. That's what's gonna take the longest. But the training isn't gonna be absolutely correct the first time, so it's hard to answer that. I just don't know. A long time. But these days, a long time for this type of work, it's not what it used to be. Like, you could do stuff on a computer really quick. Find the right tools. Yeah. Let's get this one, and then I'll get you back.
Do you think if you manipulated insect density in this space, you'd see density-dependent behaviors?
Totally. Yeah. And are they social insects or non-social? Those are all factors. Yeah. You know, this is, I repurposed my Thrips picture, but it's supposed to be Drosophila. But eventually, that generalizable space, right? What we've learned about insects can be applied to insects we haven't studied yet with a insect, right? Getting to that point of total knowledge and prediction. We need to test definitely more than Drosophila. Although I don't know. Who knows with AI? They have neurons, single neurons. Who knows? Yeah.
Can you theoretically put a plant in there?
Totally.
And see what the
I'm loving your odor talk and that stuff. So these are vacuum pumps that we made to basically push or pull air. That's what they look like now. It's like a 3D printed kit case, and then you put just a vacuum that you can buy on. It's a mini vacuum. I think you turned me onto them a while ago, and it just pumps air in a direction, a circular. So the idea here is it'll push the essential oil odors into it, but yeah, you can put a whole plant and push just that chemistry. My current thinking on the cages is going to be like a hamster ball. Just a nice spherical thing. But we're in troubleshooting. So we got to find a way that every millimeter, every volume, the whole thing is visible, right? No, nothing can be out. So anything happening in the container is being put into data, basically, right? Yeah. Oh, sorry, let me get you after. You had a question too.
Oh yeah, I was just wondering, do you already have this computer system, or?
Well, we just got the
developing
funding
it?
for it, so yes.
You have the funding for it, but then somebody still has to like develop it. I guess like.
Well, it's just a computer, you know. There are companies that make the.
Okay.
So it's on NVIDIA GPUs. NVIDIA has GPUs, graphical processing.
Okay.
Yeah. No. The technology to do this exists. It exists. Yeah. Yeah.
Instead of doing exposure to the plant, exposure to the essential oils separately, run the experiment with exposure to both to see which ones is actually preventing it from detecting that the plant is there, so it can't follow the host.
Yeah. You know, a lot of that gotta test them at the same time stuff kind of goes out the window with this once you've tested everything enough times, right? Is my current thinking, but I hear you, because we don't have the capacity to test every single possible test scenario all at once. But I hear you. The statistician in me hears you and the concern there. Yeah. I have totally forgot to ask all the questions again, so sorry everyone online. I hope you're gaining something.
So what size are those chambers that you're, because to me, like, one thing, how realistic do you think this setup is? Because you're like really concentrating the others within those chambers. So in real world, you know, that doesn't happen unless you like saturate the environment with another. So
yeah,
I'm trying like to put it in more like in realistic.
Totally. Well, you know, we can manipulate to the individual molecule. We can have like individual components of essential oils with our mass spec team pulled out, and then that concentration we just, you know, dilute to a certain amount and then apply it at a particular air rate, and it's circulating. So we'll monitor coming in, coming out concentrations, along with the idea of death volatiles or behavior volatiles that the insects are reacting with. So with that level of information, you can do a lot with, even if the concentrations aren't necessarily natural. What we could do is, after we've trained it, make predictions about, okay, with a lower concentration what could happen, based on what it knows about that chemical and every other chemical.
Do you plan to do also like after exposure, like to look at the electroantenograph, for example, like see how, you know, what you said, like the antenna habituating to the smell?
I love that. That and, you know, flash freezing them and doing neural histology. What's active now and now? Like we could freeze in that moment, and that's with people in the department, DSU. I'm in a neuroscience department, if anyone was wondering, but you know. Other questions?
So when you're talking about stuff like this, you've got fancy supercomputers monitoring thousands of bugs and all these little things, and it's so complex that we have a lot of questions. How do you dilute that down into something that like the road can understand that? We do all this fancy stuff, but here's what you need to know.
Yeah, it will be in my mind, a system that is deployed on the field, and all of this complicated neural network stuff will be programmed into a technology with a sensor on it. So they wouldn't have to know any of that. We would just say, this is going to use natural molecules to manage pests in the moment for you, and learn from them when it gets to that scale. You know, it'll be in a way that it'll keep learning. But
so you're using melanogaster as a proof of concept
to start.
Yeah, to start.
To show that
it works.
A neural network can be trained in this way on behavior. So everyone's heard large language models, and there's image stuff now. Video is kind of the edge of AI right now in terms of tools that are available. So there's definitely a Wild West feel here of making the code that makes this happen. And I do all my work in R, just so everyone's aware. Yes?
Before doing this, do you need to train the AI to actually, like using an electroentomogram for the AI to actually know? Because first twitch is the first sign of behavior. That doesn't mean it's the first sign of them like receiving the chemical. So do you first need to actually make sure that the insect receives the chemical like in different timing?
I wonder. I do. I wonder. You know, there's a lot of ways that this flips the notion upside down, because it's learning the behavior and then thinking about the published information about the organism in neural space. It's not monitoring that. So training it on normal behavior opens up the ability to pinpoint neurons when we're testing abnormal behavior, right? But you would still, and I take your word on this, you would still need to, for us as scientists, confirm that neuron that we thought, because it's black box at that point, right? The AI may know, no, it's that neuron, but we have to confirm, especially if we're going to use medicine to interact with what it's hypothesizing. Which means, yeah, we would freeze and do that, yeah.
So what sample size or like incidence would you need to validate a particular behavior of like when you're looking at something like first twitch? Like how many do you need? Like 10 million before it learns that as a concrete learned behavior, or like do you have an idea?
It will.
Yet? We'll see.
It will, as we've talked about, dynamic. This behavior, insects learn and adapt. So will the AI. So it is never like definite, right? It's always, I mean, that behavior may be associated with the likelihood that this is going to happen next, but it's still monitoring because there's always going to be variation. And so it'll update that trained variable slightly different. So in neural network space, it's basically an equation, really complex series of equations that push things through to the next logic. And it's a series of likelihoods, right? And so that is very adaptable itself. And that's the idea, is it won't be totally settled, right? It'll always be able to rethink.
But it still needs like some sort of an example though. Like how many does it need for it to be like, okay, so here is my space of anything that happens in between these things. I can kind of allude that this is going to be the behavior. But like how many samples do you have, or how many incidents of individuals do you need to have that sample size, to have variation that
a lot?
Okay.
Totally a lot.
Yeah, because I would think it would be like a ton.
Yeah,
a ton.
And you know, in the same way that we're looking at, you know, the same chemical across different plant species and having a mortality effect, that same sort of, the same behavior is happening with different treatments, sort of trend would be an equation that can be updated and modified by it. But there are going to be cutoffs, like at this point, this chemical is still working. Yeah. But it's always going to be tested. So y'all have given me so much to think about and expand on.
Yeah, this is kind of like to be continued.
I think so too.
Kasha needs to be convinced, I think.
I don't blame you.
I'm just trying to wrap my head around it. Not great with technology. I want to see it.
So this is more of like a real life application question. When you apply these chemicals to the actual like crops for like sustainable pesticides, is there like any concern about maybe the odors of these of the plants that you're using affecting like the marketability of the crops?
Only in the case where they're like having, what is it, phytotoxic effects. So aesthetically, well, melting the plant. Like with cinnamon, it was doing more damage than sweet mint was.
Right. I guess I was thinking
Flavor and whatnot.
Yeah, the actual like smell of
It washes off.
Okay.
Yeah. From my experience, there definitely was a garlic smell to some of the garlic. So we'll leave that one open. There could be some improvements that could be made. You know, that's a matter of putting another chemical in it that helps it wash off more cleanly. So it sticks on the surface of, say, the leaf that you've applied it to, but doesn't actually interact with anything. And so it can release. There are a number of chemicals that they use for that. You know, stuff like preservatives and whatnot. But just stuff that keeps it stuck, but not touching the leaf, right? The actual organism.
So I guess, sorry, a
Surfactant.
follow-up question, it would be like, how do you make sure that that doesn't cross a line into becoming more of a synthetic pesticide rather than natural? Just because like it's primarily made out of the oils that come out of the plants?
I think that's a conversation we should continue having.
I think, like based on my experience, because we're trying to register some volatile compounds with the company, EPA treats volatiles like biopesticides. So you have to go through the whole registration as if it was an insecticide. So you have to have tolerances. So you have to look at residues. So the company has to invest on a lot of these, tests to look at whether there's any residues or phytotoxicity.
Right.
We're trying to change kind of like the mentality with EPA and look at it differently, but we'll see.
Keep it up. Keep doing it. Thank you. Yeah.
Yes, I have a question like 20 years down the line. When it comes to like the application and the feasibility for growers to adopt this method, so you mentioned it would be something that is a computer system in the field basically that's already learning and continuing to learn that behavior, and that computer is then telling it when to spritz or what to spritz. So like, what is the manpower that would be required? What sort of like acreage would you need to put this across? How how many individuals would you need to start replacing these individual like spritzer systems that are in place? I mean, because that's like a lot of work. Like if you think about somebody who's got like 300 acres and they got to go every 10 feet and like put a new spritzer in there. I mean, I don't want to be that worker.
We're talking 20 years?
Is it more than 20 years? 40 years?
No, 20 years is fine. I was going to even say like 10.
Okay.
10 if we want to talk. I see drones are all over, and they're getting smaller and smaller. So monitoring, we already have the technology to monitor at whatever scale, and I don't know the exact spacing, but stuff that you put a cartridge of the essential oil or whatever the like 8 most effective high impact phytochemicals are that are diverse, and then it's maybe like a little flying drone or robot.
This sounds so expensive.
You'd be surprised, especially compared to the huge machinery that is subsidized at the scale we're talking about for agriculture. You'd be surprised, and especially the costs associated with the inputs we currently do, not just direct but indirect ecological impacts. I think it'd be saving money, and it's like a one molecule, right? Is what it could apply instead of drench your whole field.
Yeah.
Because the calendar. So that's where my thinking goes, but there's so many different ways to apply it at scale these days. It's more about another direction I'm thinking with this biological behavioral brain, machine brain, is integrating into like NASA technology as they explore to look for signs of biological activity on other planets, stuff like that. So collecting the data with the sensors and optics we have now is getting cheap. It's getting totally cheap. In robots, it's also getting cheap. Drones these days, like, you could put the canister that sprays the thing on it. I could do that today. But yeah, it might be low cost prohibitive now, but you know, showing its efficacy in that scale, it becomes obvious. Yeah. Right?
Chase, we have to stop here. But thank you very much. That's things to think about.
Thank you. Thank you, everyone. Sorry I did not repeat all the questions.
I guess there's no chat. There's nothing in the chat. So.
Okay. So just end meeting.